Hello students, in the last class I explained about the packaging stage of Prophase-I of Meiosis-I. Now in this class let's start with the next stage of Prophase-I, that is Diplotene. Okay, so during this Diplotene, Synaptonemal complex starts disintegrating. Synaptonemal complex undergoes dissolution. Complex undergoes dissolution. So what is this synaptonemal complex and what is its functions? One function of synaptonemal complex is it helps in pairing or synapses of homologous chromosomes which occurs during zygotene stage. And other function of this synaptonemal complex is to stabilize these homologous pair of chromosomes until the process of crossing over gets completed during packaging stage. So by the end of packaging stage, so this is cell membrane of the animal cell, this is nuclear membrane. These are two pairs of homologous chromosomes. So these two pairs of homologous chromosomes have paired with one another during zygotene with the help of this synaptonemal complex, and during packaging stage they have undergone crossing over, that is the exchange of genetic material between the non-sister chromatids of homologous chromosomes has been completed. So the two functions of synaptonemal complex have been completed. So and hence in the next stage, that is in the Diplotene stage, the synaptonemal complex starts undergoing dissolution. So when it undergoes dissolution, now both of these chromosomes present in this homologous pair start separating from one another. They tend to separate from one another and they get separate from one another throughout their length except at the point of crossing over. So the point where these chromosomes, homologous pair of chromosomes remain attached are called chiasmata. So the points where these homologous pairs of chromosomes remain attached after dissolution of synaptonemal complex are called as chiasmata. Now on the basis of position of chiasmata and the number of chiasmata, these homologous pairs of chromosomes appear in different shapes. Like X-like, generally they appear in X shape. So they may be X-like or ring-like or chain-like. So let's say how this X shape is formed. Let's see how this X shape is formed. So this is one chromosome having two chromatids, and this is another chromosome having two chromatids, and both of these chromosomes are homologous to each other, and this is the point of chiasmata, or this is the point where the crossing over has been occurred. So it is giving X-like appearance. So here again these are two chromosomes, homologous pair of chromosomes. This is one chromosome having two chromatids. This is the another chromosome having two chromatids. Both are homologous to each other. Here chiasmata are present at two positions, which is giving ring-like appearance of these homologous pairs of chromosomes. Next, in this here also there are two chromosomes. This is one chromosome. This is another chromosome. These are two chromatids of one chromosome. These are two chromatids of another chromosome of this homologous pair. So here at how many sites chiasmata are present? 3 sites chiasmata are present. So based on the position, on the basis of position of chiasmata and the number of chiasmata, these homologous pairs of chromosomes appear in different shapes. Okay, so what happens, and during this Diplotene stage only, after dissolution of this synaptonemal complex, both of these chromosomes, they tend to separate and get separated also except at the point of crossing over, that is chiasmata. Now this chiasmata starts terminalization, starts undergoing terminalization. That means at this point chiasmata are present. At this point chiasmata are present. At this point chiasmata are present and they start moving towards the ends of the chromosomes. That is called terminalization of chiasmata. So at first both of these chromosomes are like this. When this synaptonemal complex undergoes dissolution, they start repelling from one another. Repelling like this, they will try to separate from one another, so that the chiasmata which is present over here and here comes to terminal points of the chromosomes. So that is called terminalization of chiasmata. So this terminalization of chiasmata occurs because of repulsion of chromosomes and even because of the tight condensation of chromosomes. That is called terminalization. Movement of this chiasmata from the center position or from the mid position towards the ends of the chromosomes is called as terminalization of chiasmata, which occurs because of repulsion of chromosomes present in this homologous pair and also because of the tight condensation of the chromosomes present in this homologous pair of chromosomes. So, this terminalization of chiasmata starts during Diplotene stage, but this terminalization will be completed by the end of Diakinesis, that is the next stage of Prophase-I. So, terminalization of chiasmata starts during Diplotene stage. So, these are all the changes that occur inside the nucleus of the cell during this Diplotene stage. So, now the changes that occur inside the cytoplasm is the centrosomes or which have developed into asters. Already they started moving away from one another and they move further away from one another. Okay, I would like to discuss one important point in this Diplotene stage, that is in some vertebrates, in oocytes of some vertebrates, this Diplotene stage lasts for months to years. In oocytes of some vertebrates, this Diplotene stage lasts for months to years. It lasts for months to years. So this prolonged Diplotene stage which lasts for months to years is called as Dictyotene stage. During this Dictyotene stage, chromosomes at some part of the chromosomes, these homologous chromosomes, show decondensation. So during this Dictyotene stage, at certain parts of these homologous chromosomes undergo decondensation where genes are present. Undergo decondensation. Chromosomes undergo decondensation where genes are present. Now these genes undergo transcription to form mRNA. Now this mRNA which is formed undergoes translation to form protein. Okay, now this protein is nothing but it is a yolk of egg. Yolk present inside the egg. So generally in most of the animals, egg consists of yolk, and this yolk of egg which is present inside the egg is used in the development of embryo. It is used for the development of embryo. Okay, so this is about Dictyotene stage. Now let's see the condition of homologous pairs of chromosomes which are under this Dictyotene stage. Homologous pairs of chromosomes. Okay, so this is a pair of homologous chromosomes. This is one chromosome and this is another chromosome. Both of these chromosomes are homologous to each other and this is the point of chiasmata. And both of these chromosomes are homologous to each other. Right. Now, at certain points, some points, these chromosomes show decondensation forming loop-like structures. Loops like this. So all these loops, in these loops only genes are present. These genes get expressed, that means they undergo transcription to form mRNA. That mRNA undergoes transcription to form protein. That protein is nothing but egg of yolk which is used in the development of embryo in future. So the points where this decondensation of chromosomes occur, loops will be formed. So these chromosomes along with loops appear in the form of, gives a brush-like appearance. So These chromosomes are called as Lampbrush chromosomes. Now these are called as Lampbrush chromosomes as they are giving brush-like appearance. And as they are larger in size because of decondensation, they are larger in size. As they are larger in size, they are also called as giant chromosomes. Giant chromosomes. Okay. So this is about the condition of homologous pair of chromosomes during Diplotene stage, that too during Dictyotene stage. Here I would like to give one example of this Dictyotene stage. So in case of human females, oocytes of all human females reach this Diplotene stage at the 5th month of fetus. After reaching the Diplotene stage at the 5th month of fetus, this Diplotene stage is suspended. 5th month of fetus. This Diplotene stage is suspended and the meiosis will be resumed during the time of ovulation or at the time of puberty. So meiosis will be resumed. That means meiosis is suspended. Where it is suspended? During this Diplotene stage. So in case of all human females, oocytes reach Diplotene stage at the 5th month of their fetus and meiosis. And during this 5th month of the fetus, they are suspended. Their meiotic phase is suspended during this Diplotene stage. And this meiosis will be resumed at the time of puberty, that is at the time of ovulation. Resumed at the time of puberty or at the time of ovulation. Puberty or ovulation. So this is all about Diplotene stage, which lasts for months to years in case of the oocytes of some vertebrates, which is also called as Dictyotene stage. So I just give a quick recap of this Diplotene stage. So during Diplotene stage, the synaptonemal complex undergoes dissolution, due to which the homologous pairs of chromosomes, they tend to separate from one another and they get separated from one another throughout their length except at the site of crossing over. The sites where they remain attached after dissolution of the synaptonemal complex are called as chiasmata. These are called as chiasmata. Right, based on the position and even number of chiasmata present in these homologous pairs of chromosomes, they give X-shaped appearance or ring-shaped appearance or chain-shaped appearance. Generally, they are in the form of X-shape. Okay. So in some oocytes of some vertebrates, this Diplotene stage lasts for months to years. So this prolonged Diplotene stage is called as Dictyotene stage. During this Dictyotene stage, this homologous pairs of chromosomes show condensation at certain points where genes are present. Okay, these genes undergo transcription and form mRNA. This mRNA undergoes transcription and forms protein. This protein is nothing but egg of the yolk, which is utilized during the development of embryo. Okay, now the condition of homologous chromosomes during this Dictyotene stage. So its condition is, so these two are the homologous chromosomes and this is the point of crossing over, chiasmata. And at certain points it is showing decondensation, so it is forming loops at these points. These chromosomes, homologous pair of chromosomes along with loops gives an appearance of brush. So these chromosomes are called as Lampbrush chromosomes, and as they are larger in size, they are also called as giant chromosomes. So for example, one example of this Dictyotene stage is, in case of all human females, oocytes reach this Diplotene stage at the 5th month of the fetus, and meiosis is suspended at this Diplotene stage for months to years, and then again this meiosis will be resumed back when at the time of puberty or ovulation. So this is all about Diplotene stage. Next, let's start with diakinesis. So now next phase of prophase-I, that is diakinesis. This diakinesis is the last phase of prophase-I. Now let's see what happens during this diakinesis. So during this diakinesis, terminalization of chiasmata will be completed. Actually, terminalization of chiasmata starts during Diplotene stage and it is completed. Terminalization of chiasmata will be completed by the end of diakinesis. So what is this terminalization of chiasmata? That is movement or slipping off of the chiasmata from center position towards the ends of the homologous pairs of chromosomes is called as terminalization. So this terminalization occurs because of repulsion between the chromosomes due to the dissolution of synaptonemal complex and due to tight condensation of chromosomes. This terminalization of chiasmata occurs, which is started during Diplotene stage and which gets completed during diakinesis. And during this diakinesis, nucleolus present inside the nucleus disappears, and all other cell organelles like Golgi bodies, endoplasmic reticulum, mitochondria, and even chloroplast. If it is animal cell, all others chloroplasts are absent. All other cell organelles present inside the cytoplasm of the cell disappears. They disappear, and even the nuclear membrane starts disintegrating. When the nuclear membrane starts disintegrating, cytoplasm and nucleoplasm gets mixed with one another. So there will be no differentiation between cytoplasm and nucleoplasm by the end of diakinesis. So this diakinesis is representing the transition phase from prophase-I to metaphase-I. So by the end of diakinesis, formation of spindle apparatus will be initiated, and by the early metaphase-I, formation of spindle apparatus will be completed. So diakinesis, we can say it as it is a transition phase from transition of prophase-I to metaphase-I. So in the next class, let's start with metaphase-I.